did like the clown makeup. I did all that. And I'm walking through the store and people are like kind of staring. And I'm like, I'm really tired. And like I see this old lady, she kind of looks like kind of shocked. And I'm all like, is my hair not combed? You know, this is like early Saturday. So I'm like, maybe they can comb my hair. And finally, I've seen enough strange looks, thankfully it was early. I pull my phone out and turn it on photo mode and turn around, and I look like Mose on the clown. I got like red lips and I was cracking up. And I almost, the reason that reminded me, I almost did that here. I pulled my chapstick out to get ready to lecture, and I grabbed the collar. I'm like, no, not going back. Yes,
That's what I was wondering about.
oh, yeah, these are the normal life of when we have like, there's always something.
Halo files?
Okay, how do you feel about the quiz? Good. I really felt like they're really gold, right?
What was the question again?
So I do want to get on our lecture stuff today. Is that okay if we wait? Yeah.
High osmotic pressure and
I'm going to make it my priority, so we'll give you a nice one.
I didn't understand it.
I want to jump into chapter 6. Maybe like how long it is. Okay, are we ready to start?
Oh,
Halo
files.
There we go.
That, yeah, hold on.
Okay, so last time we were talking about growing different organisms, and we were talking about how they have certain temperatures.
That's the first time I never know what answers to say.
Okay, we're talking about how they have certain temperatures. We said physical and chemical requirements that they prefer. And then we were just getting into about how cells spend most of their time in interphase, whether they're prokaryotes or eukaryotes. And interphase is when they're just living their normal life. But when a cell gets a signal from their DNA that it's time to replicate, they'll start processing and getting ready while they're still in interphase. So one of the things they'll do is they'll do some DNA replication. They make a new strand and an old strand together. And then they'll make if there are cells, they'll do some, they'll make some organelles. They'll make some extra organelles, and they'll get ready to enter into cell division. So that gets us to mitosis, and remember, is mitosis for prokaryotes or eukaryotes? Eukaryotes, good. And remember, eukaryotes include things like yeast and fungi. They're going to be in the same group. So there are 4 main stages associated with our cell division process. That's going to be our prophase, metaphase, anaphase, and telophase. Remember, these are going to occur after interphase ends. We'll immediately go into prophase, and then we go quickly through these stages, and they occur one right after another. Now, one thing you want to keep track of is the order of the stage, because each stage leads to the next. So the order is important, and then knowing what the main events are at each stage. So one of the ways to keep track is to make a little mnemonic. You can say, people make amazing things, or pass me another tri-tip, or pass me another taco. I'm a foodie, so I always have like mnemonics, right? I had a teacher who said, pass me another test, and I thought, well, that's kind of lame. Taco's much better, but whatever you want to come up with, that's what you can come up with. So we'll review these in a little detail here. So the first step of mitosis itself is prophase. And when you see prophase, I want you to see that P and think plain to see. And the reason for that is because prophase is where our DNA is going to condense. And what that means is it's going to tighten up. It's going to be really tight together. And if I look at a cell that's in prophase, I'm going to be able to see the DNA appearing as threads rather than just kind of willy-nilly. So remember, in our cells, usually our DNA is relaxed. Does anyone remember what that's called when it's relaxed? It's like a throwback. If you don't, it's fine. It's called chromatin. Chromatin is my relaxed DNA. When I tighten up, that's chromosomes. Okay, so I'm going to see the chromosomes tight. I'm also going to see that the nuclear envelope starts to break down so I can get to that DNA. And I'm going to have spindle fibers form. Spindle fibers are special proteins. They're microtubules. Think of them like fishing lines. They basically extend and they grab on to the DNA. And they're going to be responsible for helping to move things where they need to go. So the microtubules are going to help move the DNA and the organelles around so that I get an even number in my old and my new cell. And then they're also going to be responsible for helping to push new elongate the cell when we get ready to go through our actual separation. Now the next stage that occurs right after that prophase is going to be metaphase. When you see M, think of middle. Okay, so this is where the chromosomes are going to line up in the middle. And hopefully this makes sense because one of the easiest ways to keep track of my chromosomes is to organize them. It's kind of like if I have all of my students spread out, there's not a lot of organization, but if we had an emergency we needed to leave quickly, we could form a line and then I could do a head count to make sure everybody's covered. That's basically what's happening here. We're in a line and the microtubules are double checking to make sure that they're all attached to some DNA. So we got it all organized. The next thing we'll also do is we'll make sure that sister chromatids are facing opposite poles. So let's explain that. Remember, a chromosome is where I've had a strand of DNA copy itself, so it looks like an X right now, okay? So in this case, I'm gonna make sure one sister is facing one direction and one sister is facing the other. Because ultimately, what's gonna happen to these X chromosomes? They're gonna get pulled apart. So what I don't wanna have happen is have them like this, because then they might both go the same way, all right? So I need to make sure that they're getting ready to separate. That brings me to my next stage, which is anaphase. When you see A, think apart. We're pulling things apart. So anaphase is where those microtubules that are attached to the DNA contract. And this is where that fishing line analogy comes in. It's kind of like reeling in your fishing rod. That's what's happening. Enzymes are pulling the microtubules tight, and as they contract, the DNA that's attached to them separates. So this is how you get sister chromosomes that are chromatids separating. They're going to move to the opposite end of the cell. Now I'm also going to have some microtubules that are going to form that are going to help push against each side so that way we can elongate the cell. So our cell's going to look a little funky for a little bit. It's going to look kind of elongated, almost like a jelly bean. It's going to look a little long, more so than normal. And then we get to telophase. And when you see telophase, think of T for 2, because we have 2 nuclei that are now present. So my DNA's been separated. It's on each side of the cells that are going to develop soon. And so my DNA is on this side, and I have organelles on this side, and perfectly replicated DNA and organelles on this side, and I'm getting ready to separate the cells physically, okay? Now, when I look at the cell at this stage, I can tell I'm in telophase because I'm going to see 2 nuclei, all right? So if I look at it through a microscope, I'll actually see the presence of 2 nuclei, which we can see there at that top. Now, the nuclear envelope is going to also start to form, and then we're going to be ready to physically split our cells. Now, this is technically the end of mitosis. So mitosis only goes through prophase, metaphase, anaphase, and telophase, and then we're done. But you might look at that and go, I think there's another step that's missing. What else do I need to do to my cells here? Split them, yeah, so I need to split them. So that's actually a separate process. That's going to be the process of cytokinesis. So for us, as soon as mitosis ends, we immediately do cytokinesis. We don't have any hesitation. But remember we talked about yeast. And I talked about how yeast do budding. So when they go through telophase, they don't necessarily go right into cytokinesis. They'll stay together, and I'll see 2 separate nuclei. They'll reform their little cell membranes and their cell walls, but they're going to still stay attached. They're not going to physically separate, okay? So cytokinesis is physical separation. For us, the way that's going to happen is we have proteins and enzymes that wrap around the cell, and they're going to create what we call a cleavage furrow. They basically bend in the middle of the cell. And the way you can think of this is like a drawstring bag. Y'all ever pulled a drawstring bag, you pull that string and it contracts? That's what's happening. So these enzymes and proteins are working together. The proteins are like microtubules, like fishing lines. The enzymes contract them and they pinch in. And when I pinch, I physically separate the 2 cells. So I'll see that cleavage furrow that's developing. It'll continue to pinch through until I pinch the cells in half. So if we put this all together, we'll start an interphase. What do I do in interphase? Yeah. yeah, I live my life and I make a copy of DNA when I'm ready to go to mitosis, right? And I make copies of organelles. So I start an interphase. Cells spend about 80% of their life in interphase. Okay, that's their dominant place. Then they're going to go into a situation where they're ready to go through mitosis. So the first stage of mitosis is prophase. When I see T, what do I think of? Plain to see. I can see my condensed chromosomes and the nuclear membrane starting to disappear. All right. Next, I start to see that I'm ready to go into metaphase. Metaphase is where I'm going to line up my chromosomes in the middle, making sure the sister chromatids are facing the opposite directions so that they're all balanced. And once I line them up in the middle, I immediately get ready to go on to anaphase. And anaphase is where I'm going to have the actual physical separation of those chromatids. So they separate, one sister goes one way, one sister goes the other. So now I'm in anaphase, and once I'm in anaphase, I am now ready to have my chromosomes start to relax. I start to reform the nuclear envelope, and I'm in telophase. When I see T, what do I think of? T for 2, yeah, so 2 nuclei are present, very visible to see. So for some for some yeast, they stop here. For our cells, this is the end of mitosis, but what do we immediately go into? Yes, exactly. So we immediately are going to go into physical separation, which is cytokinesis. All right? So is that kind of nice just to see that all balanced out? Please tell me it was nice because I spent a lot of time animating that. I thought it was really cool. I was like, oh, this is going to be fun. And then I saw your slide print out and it's kind of a mess. But you get the idea, right? Like everything's there. All right. So with this, there are certain cells in the body that are constantly going in and out of mitosis. So for example, our skin cells. Skin cells are constantly having to be repaired and they're constantly being shed. I know this is kind of gross, but like the dust in our house, most of it is old skin cells, right, that accumulate after a while. I probably have enough dust in my house right now to build a whole other clone of myself because I haven't had time to dust lately. That's probably too much information. But anyway, there you go. So skin cells are constantly being sloughed off and replaced. We get a boo-boo on our arm, it's going to replace, and those skin cells are going to come back. Other cells only undergo mitosis if they're stressed. So for example, liver cells. If I'm not bringing toxins into my body, then my liver cells aren't in a stress state. Because remember, the liver is detoxing, right? So let's say it's Super Bowl Sunday, or let's say it's Christmas, or it's a holiday. What might I be doing on those days? Drinking, yeah. Maybe I'm drinking some wine or some alcohol. Wine is good for the heart. We're going to stick with that. All right, so we drink some alcohol. It comes in, and our liver cells are detoxing for us. Great job. But that does put some pressure and some damage on them. So they're going to go through mitosis to regenerate. Okay, so our liver cells can then regenerate. There are a few cells in the human body that never regenerate. Where do those live? Yeah, in the nervous system. So unfortunately, we have some neural cells that if they're damaged, that's it. And we do have a few things that we can try. For example, spinal injuries. Sometimes maybe you've been in a motorcycle accident or you've had a spinal injury. You might lose the ability to walk because of the damage to the nerve net or the neural system. The cells there, they're so complex and so complicated they don't regenerate. So used to, if that happened, you're just out of luck. Nowadays, we have some new things that we're trying which work around stem cells. So we talked about stem cells in here before. Just briefly, stem cells are baby cells. They don't know their job yet. So if I inject stem cells near neural cells, what usually can happen is those stem cells kind of look around and see what their neighbors are, and then that's what they turn into. So if they're near nerve cells, they'll become a nerve cell. Now, it's a little bit more complex than that because not only do they have to become a nerve cell, but they have to start communicating with their neighbors. So stem cell treatments, they're not fast, they're not quick. They take a lot of procedures and processes, but they do occasionally work. So there are a lot of papers that have been written about people recovering the ability to walk or having spinal repair after like a year of therapy with stem cells. So again, it takes time, but if I have a chance, I'm gonna jump on it, right? Where are stem cells collected from mostly today? From, not necessarily embryo, from the umbilical cords. So we used to collect them from embryos, and that created a huge controversy. The concern about the whole embryo collection, so a lot of times, like for example, if you underwent IVF, you would create embryos, and then let's say you had, maybe you set up like 20 embryos, maybe you overcollected. And now you used your 2 embryos, you successfully had 2 pregnancies, maybe you were done. What do you do with the other 18? So a lot of people would donate them to science, that's where we would get stem cells. Also with natural abortions, if the baby was rejected from the body, they would be collected, and then that would be a source of stem cells. The controversy that came from stem cells, which it makes sense if you think about it, is a black market. What if we're in a situation where we don't have any money, and if I become pregnant, I can sell my embryo to be able to get stem cells? And that was what the concern was, that we would create a black market, or human trafficking would be using women to create embryos. So that's why it was shut down for a while and heavily regulated. We realize now we can get stem cells from a lot of different places, but one of the best ones is the umbilical cord. So anytime you give birth, nowadays one of the questions on the forum is, do you want to keep your umbilical cords? Sometimes certain groups like to, like Native Americans, for example, will keep the umbilical cords and they will bury them near to a special place or a shrine. But if you're not going to use yours, you can donate it to science, and that's a great place for them to be able to get a lot of stem cells. One thing I will caution you about is they'll ask you if you want to save your umbilical cord from your child, because they could use it in the future for stem cells. And they have this note on there, like if your child becomes sick and needs stem cells, they would have access to their own stem cells. Well, it sounds good in hypothesis, but the likelihood of you needing stem cells is pretty low. And what they don't tell you is you're going to be paying a storage fee for that service. So you might be paying a storage fee because the parent, you're paying it for 18 years, and then your kid might be going, I don't want to take that on at 18 and then throw it away. That was 18 years of storage fees, right? So it's always good to kind of ask. And then the other thing to keep in mind is stem cells have a really low rejection rate because they're babies. They haven't differentiated. So when we put them in somebody's body, they actually will create the right proteins and enzymes to blend in because they're being dictated to by their neighbors. They're being told what to create. So it's not like you have to have just your own. You can have pretty much anybody's stem cells. All right. If you do need your own, it turns out even old people like myself, we have stem cells. We have stem cells deep in the epidermal, actually endodermal tissue. We have stem cells in the eyes. We have stem cells in a lot of places that we can actually touch and from. So I would just caution you with that. Don't fall for the cells pitch, okay? Unless you really think there might be something in your family that makes you need to have stem cells. Okay, unfortunately, sometimes the process of mitosis gets stuck. So usually mitosis is heavily regulated. We have all sorts of enzymes present and genes that have to be turned on and off to start the process so that we don't get uncontrolled mitosis. But unfortunately, sometimes we get certain genes that are called oncogenes where it's like they're stuck. And so they go through mitosis and they create daughter cells, and then they go through mitosis immediately again and create more daughter cells, and then again, and then again. And so what happens is we start to have these cells build up, and that's what we refer to later as a tumor. Okay, so a tumor is where we've had cells with uncontrollable division. Right? Now, we mentioned before that we can find cancer cells by tagging certain types of nutrients. What do cancer cells love to eat? Sugar. Yeah, because if I'm going through mitosis all the time, I need a lot of glucose to make sure I'm getting sweet. So I'm going to be a big sugar eater. So that's how we can find cancers in a lot of patients, is by tagging glucose and seeing where it starts to concentrate. And if we see it concentrating in an area, that a lot of times can indicate that there might be a tumor present. Now, one thing that is tricky with cancer is when a tumor is genes and tells them not to grow. So in other words, if I'm a tumor and I'm developing, I'm going to inhibit other tumors from developing near me because I want all the resources. So one of the problems with removing tumors is we have to make sure that we remove them completely and any possible neighboring areas, because if I don't, what could happen once I remove that main one? It can metastasize. Other tumors that were really small, maybe I didn't detect them, they were like being told to wait, they just had their master tumor removed and now they're gonna start growing. So that's one of the things you have to watch for with cancer. So it gets a little tricky with trying to deal with cancerous agents. Other types of replication would be things like cnidiospores with mold. So this is a close-up of mold. Aren't they cute? Don't they look like little lollipops? I think they're adorable, right? So this is mold. When we see it on our bread, it doesn't look so cute. If we got a microscope though, this is what it would look like. All of these little circles are little cnidiospores. So the way that these form, the cells go through mitosis, they create a cister, but they're kind of in a spore form. Now remember, it's kind of like an endospore. They're not actively growing yet, they're slumbering, okay? And then what's gonna happen is when these mature, when they get big enough, these little individual pieces can break off. And they can scatter in things like wind currents, air currents. They can scatter from any kind of shaking or movement. And when that happens, one of those will land, one of those spores will land, and it'll start growing again. This is what mold looks like on a petri dish. And it's really hard to know how many individual colonies are actually here. If I look at this, how many would I think are there? 3. I see 3 main ones. But in reality, this was probably about 50 little tiny dots when they first started, and then they all merged together. So counting molds can be a little tricky. You have to catch them when they're young if you want to actually get like a colony count, because the older they get, the more they spread, and they just kind of encompass everybody. Okay, it's kind of like urban sprawl. They'll just keep spreading to the point where I can't even tell anymore where Orchid and Santa Maria are separated. Have y'all been to Orchid lately? It's like blown up out there, right? It's like I drove through there not too long ago. I was like, oh my gosh, there's so many houses out here. It's like crazy. This is a picture of the bad mold. So luckily we don't have this mold very often here in California. The reason for that is because this bad mold, sometimes called the sick mold, its real name is Ch.6. It prefers to live in communities with a lot of humidity. So we don't have a very humid environment here. So because of that, we don't see too much stachy here. But if you go to the south, Ch.6 is very, very prominent. If you see mold in your bathroom here, 9 times out of 10 it's probably aspergillus, which is harmless. You spray with some bleach and you can kill it. It's pretty easy to get rid of. It'll come back because it has spores, and the spores are resistant to a lot of cleaners, but it's not going to make you sick. Ch.6 makes you sick because it spreads really quickly and it's a really high spore producer. So what that means is when I see it, there's millions of spores in the air. So if I breathe this in, those spores are going into my lungs. They're going into my circulatory system. Now, I might not have the mold ever grow. It might get, immediately killed by my immune system and taken out. But just by the sheer number of spores, it can make me feel tired. It can make me feel like I have a headache. It can make me feel achy because my immune system is trying to fight them off. So that's why I get sick mold syndrome. If I keep getting exposed to this high number, it could lead to where I'm actually getting sick, like lung infections, respiratory infections that could result in mold infection. We don't want mold infections. If we get a mold infection, they're hard to treat. Why? They're eukaryotes. Good. They're just like us. So anything I target on a mold, I potentially am hurting myself. So trying to kill off mold with antibiotics is kind of a delicate balance. We're going to have side effects, guaranteed, with the medications used. But we have to go to the edge of those side effects to be able to slow these guys down and grow. And then we have to keep pushing. So Mold treatments, they take a while, and they usually have some side effects associated. Okay, so when I'm growing my bacteria in a laboratory setting, or when I'm trying to determine how much bacteria or mold I have a couple of different ways that I can determine if growth is present. So one of the ways that I can chart my bacterial growth is to use something called a bacterial growth curve. Now, this is just kind of a general chart showing how bacteria reacts when it's moved from one media to a new media, or one environment to a new environment. So when I add bacteria, let's say today we set up some broths. When I first add the bacteria, I'm going to have my stage 1, it's referred to as the lag phase. And what's happening here is the bacteria are figuring out what nutrients are present and what kind of environment they're in. You can picture this. This would be like you're walking to your car at the end of class and a big hand grabs you and drops you off somewhere else. That's what's happening when we're inoculating our bacteria. The first thing you're going to do is you're going to get up and be like, what just happened? And then you're going to try to figure out where you're at. And then what resources you have. That's what the bacteria are doing here. So it's called the lag phase because they're not reproducing yet. They're just trying to figure out what happened. Okay? Then, after a little bit of time goes by, they realize, okay, the environment's good, there's nutrients here, I can start creating my sister cells, and we're going to get what's called the log phase. The log phase is where we're going to have a lot of rapid growth. So really quickly, logarithmic growth, doubling every 15 to 20 minutes, getting lots and lots of cells. After a little bit more time goes by, I'm going to hit the stationary phase. The stationary phase is where I'm still reproducing, I'm making new cells, but the original cells that were there are aging. They're starting to die off. We're starting to have a little bit more competition for resources, for nutrients. So we're getting a little more stress starting to be introduced. It's kind of like if you move to a neighborhood nowadays, there's like a lot of people fighting for buying houses, right? It's really competitive. That's kind of how this is. There's just a lot of competition for nutrients. Our population numbers are staying about the same because the number of cells aging and dying are being replaced by the new babies that are being born, but still we're having to find a little more. That brings us to the last stage, which is the death phase. And the death phase is because a broth or a plate only has a certain number of nutrients available. So after my population has exploded, after I've had all these cells live together, they're now fighting for resources, there's not enough food, there's not enough space, I'm getting lots of competition. So now the number of cells dying and that are stressed and that are making endospores is really high compared to the number of cells being born. Okay, so I've kind of like gone full circle here. Now, if I want to know how many cells are in a sample, I have a lot of tricks that I can use. This is called a serial dilution, and this is actually used by food companies when they sell and distribute different food products to Americans and to other countries. So the way this works is, let's say I work at Foster Farms, and I'm selling chickens. And I want to know how many bacterial cells are in a gram of my chicken that I'm getting ready to sell. Well, one of the ways that I can determine this is I can take that gram of chicken, I can dice it up, maybe I'll put it in a blender or something to separate it, make it really, really tiny. And then I'll add it to a set volume of a dilution. So I'll have like 9 mils in here, I'll add 1 mil of my sample, so I get a total of 10 mils, which is a 1 to 10 dilution. I let that sample sit, but I know I'm going to have a lot of bacteria. Does this chicken have a lot of bacteria? Yeah, it naturally does. So I'm going to take another mil from this tube to go to this tube, and now I have a 1 in 100 dilution. And then I'm going to take a mil from this tube to this tube, a 1 in 1,000. Basically, each time I add a tube, I'm adding another 0 to my dilutions. Okay? Now, then, at the end of this process, with the last 3 tubes that I make, I'm going to plate them. So I'll take a little sample and put it on a petri dish. So the last 3 seeds I made. I come in the next day and I count the number of cells, so the colonies that are present, divided by the volume that I plated and the final dilution, and that's going to tell me the number of colony-forming units that were in my original sample. Okay? So every food manufacturer uses this because if I'm going to sell food, there is a certain number of bacteria that is determined to be safe. Is our food ever sterile? No, it is not sterile. There is always bacteria present. But every food item out there has a set number of cells that says, okay, you can sell this chicken as long as the cells aren't over this range. So this is how they confirm. Okay, so they would do a test like this. So here's the process showing how they would get to their plates. So one of the ways they could do, as I mentioned, they add the sample to the surface of the plate, spread it out, and then count colonies the next day. That tends to be a very easy, fast way. This is actually a process we're going to do in our lab. We're going to do some cereal dilutions. We're going to test some milk. We're going to test some hot dogs, and we're going to test some sprouts. So we're going to be some food testers. We're going to see what grows. So I might change you for life. We'll see. All right? Ah, so I got to get the board here. All right. Over here, this is another way I can do the same test. Instead of adding the inoculum to the surface of a plate, instead I can add the inoculum, the liquid, to an empty petri dish and then pour media on top and swirl it. Now, I did try to have my students do this technique. It was a dismal failure. Okay, and here's why. There's a little bit of trickery with this technique. If I pour the media in while it's too hot, what's going to happen to my bacteria? I'm going to kill it. Yeah. So when we tried to do this in the lab, we just couldn't seem to find that perfect temperature. To stay liquid, remember I have to be about 40 degrees Celsius. If I went down to 38, I would start to solidify. So when students were pouring, they'd get chunks. We don't want chunks. But a lot of times what would happen is students would pour directly out of the heat. It was too hot. It would kill the bacteria. So we don't do this process anymore. We do the spread plate. But this way does work if you work in a lab where they have like heated baths that are perfectly temperate that you can come from. We're not that fancy, so we do the spread plate. All right. Now, when we're done then, the samples that we set up should have a countable range. So this is kind of showing you what you would see. The first dilution would be full of bacteria, way too many to count. Even the second and third usually are too many to count. But as I start to get up to fourth and fifth dilutions, I start to see isolated colonies. I've diluted them enough. What I need to do is I need to find a plate that has between 25 and 250 colonies. The reason for that is there's been a lot of statistical analysis done, and this is considered to be the golden number, 25 to 250. If I'm below 25, statistically, my number is invalid. So if I'm at 20, that's not enough colonies to ensure that I got a right sample size. Maybe something was off with my sampling. Maybe something was off with my technique. So I need to have at least 25. If I get over 250, maybe I wasn't diluted enough. So I have this golden window that I try to shoot for. So 25 to 250 is the golden range. Now this is straight from the FDA website. If you go to a food company, sometimes they kind of change it. Sometimes they'll say 30 to 300. Sometimes they'll say 40 to 400. I'm going by the regulations, by USDA and the FDA. Okay, another way that I can find out if bacteria are present is to do a filtration method. This is a really handy way to see if I have bacteria in, let's say, like a water sample or milk, like in a liquid sample. So the way I do this method is I have these vacuum pumps hooked up to these containers, and I have a petri dish nearby, and at the bottom of this vacuum pump container, I have a filter paper. Now the filter paper allows everything to pass through that's a certain cell size, but it'll trap any bacterial cells. Okay, so if I'm a bacteria cell, I'm too big to fit through the filter, so I get stuck on the surface. So what I do is after filtering my water, for example, I then unscrew this container, and with sterilized tweezers, I remove the filter and I put it onto a petri dish. And then I wait to see what grows. So that's what we're doing here. So we're gonna do this in lab too. We'll remove filter papers and put them onto petri dishes to see what grows. It's kind of a cool thing. Now, another way that we can determine how many bacteria are present is to do counting. This is a very tedious process. Imagine your microscopes. Y'all remember your microscopes. You're staring through your microscope. You would use a special slide that has grids on it, and you would sit there and count by looking through your microscope how many cells are present. We used to tease that we would only make the lab techs that weren't listening do this kind of counting. Right? This was like your punishment. It's like if you're not going to listen or you set up a sample wrong, go count cells. All right? Because that was a punishment. Trust me, doing a filter for doing growth on plates are much better than sitting there trying to count. Okay, another way we can determine how many bacteria are present is to use statistical analysis. The military actually showed us this one, and microbiologists loved it and embraced it. This is a way we can tell if we have bacteria in drinking water, and it's a really quick process. This uses something called an MPN table, which is at the top, meaning most probable number. You can see that. Now the way this works is I pull a sample of water and I set up 5 tubes. And originally all of my tubes are dark green, or they could be red depending on what pH indicator I use. And I let them sit and incubate for 24 hours. I then come in the next day and I look to see if I see any color change. If I see a color change, that indicates that I have bacteria growing. Then I compare how many tubes were positive versus how many tubes were negative. So in this case, 3 out of my 5 tubes were positive. So I compare that data with an MPN chart. It's a set value. And I just find the number of positives I had and look at that table, and it will tell me how many bacteria I had in my water. Now, when we do this test, we usually run it in triplicate. So we'd have 5 test tubes set up, but I would set that up 3 times. So I would have 5, 5, and 5, which means I'll have 15 tubes total. Everybody with me on that? So here's a picture of your MPN test. On that example, I said that I had 3 that were positive, right? I had 2 that were negative. Let's see, out of my 5, let's see, I'm trying to see if I have one that's example here. So 3 that were positive, 2 that were positive maybe in the next run, 5 that were positive in the first run. So this is test 1, test 2, test 3. That tells me I have about 140 bacteria per 100 mils. So 140 colony forming units. So that tells me I have a lot of bacteria. I don't want to drink that water. Everybody see how that works? So all I'm doing is I'm coming in and reading how many positives I have in each set. So in this set there would be 3. In that other set there was 2. In another set there was 5. Does that make sense? And I'm just comparing them with units. Now if I was to ask you an exam question on this, I would give you the table and I would tell you what you were seeing for your positives. I would say you had 3 positives, 2 negatives, 4 positives, and ask you to interpret on a table. You do not have to memorize it. It's about understanding the text. Does that make sense? Think you could do that? If I gave you the table? Yeah, I feel lots enough. Okay, good. Okay, so those are all ways that we can determine if bacteria are present just by how they grow. We can also do quick and dirty indirect ways. This is where we're not having to wait for any kind of growth. We're just going to do a quick analysis. So the goods about these is there's no incubation time, there's no waiting. I get a quick turnaround. The bad is it's just telling me that cells are present. It's not telling me if the cells are actually living, if they're actually reproducing. It's just saying they're present. So for example, if I take a water sample and I look at it with a spectrophotometer. Spectrophotometer shines light through my water sample. I measure how much light passes through and how much bounces back. I get a turbidity level, okay? So when I'm shining that light through, it's telling me that there's some debris present and I get a turbidity reading. It doesn't tell me that all of the debris is cellular. In a water sample, what other kind of debris could I have in there? Dirt. Yeah, I could have dirt bouncing back. So it's not as accurate, but it's still a quick and dirty. It's just a quick way to tell how many cells are present or how much stuff is present. Now, another way that we can use this is to do a visual test. This is called a McFarland sample or a McFarland standard, and we use these in microlabs around the world to make sure that we're starting with the same inoculum. So let's kind of see how this works. We sell these standards, you buy them from these companies, and inside of them they have suspended latex. And when you look at them, notice that they have different levels of cloudiness. Do you see how they get progressively cloudier as we go to the end? So each of these is their own McFarland standard. This is a standard 2, this is a standard 3, this is a 1, and this is a 0.5. Now the reason this matters is if I'm doing, let's say, antibiotic testing. We showed those pictures last time of the antibiotic plates that have the big halos. The reason labs want to have a general start is, let's say I'm lab A and I use a 0.5 McFarland to test my antibiotic. And I'm going to compare my results with lab B, but unbeknownst to me, lab B used a McFarland that was a 3. Is there going to be a difference? Yeah, huge difference, right? I have a lot more cells that my antibiotic has to kill, so I'm going to have different results. So McFarlands allow labs worldwide to start with the same number of cells. Now, it's not perfection, we're just looking for a range. So for example, here, our 0.5 McFarland, the range on it is about 1 times 10 to the 7th to 1 times 10 to the 8th. So the way I would do this is in the lab. I would have a McFarland sample. I would get a broth, just like what I hand out to you, like nutrient broth. I'd get my loop and I'd scoop up some bacteria and add it to the broth and then vortex or shake it until I matched the turbidity of the McFarland standard. Everybody with me on that? So I would keep adding until it was the same turbidity. And the way I can tell what the turbidity is I use these little, they're called Wickham cards, that show me with lines how opaque they are. So this is just a quick and dirty, it's like a real fast way to set things up. But when I'm done, I'm making sure that I'm starting with about the same range as somebody else. Does that make sense? So it's just a way to regulate, to make sure that we're starting with the same range. Now I know, looking at this 10 million versus 100 million cells, that's huge, that's a lot of difference. But it's better than like 100 million versus, I don't know, 100 billion, okay, where we can get things really quick. Does that make sense how it's used? Okay. So if we were to compare direct and indirect methods, direct methods again is where I'm growing or I'm doing some sort of a physical count on living, actively living cells. So plate counts, filtration, emptying, direct microscopic count, I'm counting or looking at living cells. Indirect are the quick and dirties. Turbidity, McFarland standards. I'm looking for some sort of metabolic activity. It could be like an enzyme test. Like I could do a quick lactose strip paper to see if lactose is present, something along those lines. Okay? That's going to be the way I can tell if I'm growing my gut. Okay, so let's do our checkpoint here. Why is it difficult to measure the growth of mold when you're looking at a plate? Remember my example of the mold. How many colonies did we think we could see? 3. But could there have been more?
Yes.
Yeah. So the reason for that is because filamentous colonies are hard to count. They merge together. So I'll see 3, but there could actually be 50, 60, I don't know. They all merge together. Direct methods usually require an incubation time. Why is this not always feasible when I'm analyzing food? So why would I want to avoid an incubation time? What's the downfall of an incubation time? Yeah, I have to wait. Different time. Yeah. So if I'm a manufacturer, time is product is safe, they'll release it to send it out, and then they'll continue to do incubation tests. If their initial numbers were off, and after their incubation tests, they find that they have a lot more bacteria, what does a food company have to do? A recall. Yeah. So this is the tricky point for a lot of food companies. Do we rush and release and then recall with egg on our face? Or do we take our time and release but have a shorter time to actually get product? And it's something that every manufacturing company has to kind of juggle. And there's no one right answer for every food out there. That's why there's different expiration times on every product that's on the shelf. Okay, so that comes from food analysis and how likely there is to see bacteria, and then over time how that bacteria can grow. Okay, so food may expire before testing is complete. That was a nice short way to say what I just said. All right, BSL, what does BSL stand for? Biosafety level. What BSL is influenza? 2. Yeah, so BSL is a 2. There is a rare strain, thankfully we haven't seen it in public since the 1918s and 1920s, called Spanish influenza. First of all, this is really an unfair name. When you hear Spanish influenza, which country do you think was the first to get it? Spain. That's not true. Spain was the first that actually acknowledged it. And they got punished by having a flu named after them. So all of the countries were fighting Spanish influenza. Spain was the first one that said, I think there's an outbreak, and they went, you know what, you're right, we're naming it after you. And of course, yeah, they were trying to do the right thing and not punish. So Spanish influenza is a really crazy strain of flu. It actually causes pneumonia, so it causes a lot of fluid in the lungs. It has a very high mortality rate because you effectively drown. You die in your own fluids. So, Spanish influenza swept the nation. It was our last pandemic before COVID, and it killed a lot of people. It, just like COVID, would wipe out entire communities. So it was just very similar to COVID. The difference, COVID had a very low death rate compared to Spanish influenza. Spanish influenza was actually more contagious and even a higher death rate. So, luckily for us, Spanish influenza kind of burned its way out. Some people were naturally resistant, it kind of disappeared. We do have samples of Spanish influenza, and let me tell you, it is just as nasty today as it was back then. The CDC in 2004, I think it was, they had some trainees that were going through to study it. They meant to grow H1N1, which was swine flu. This actually would have been around 2011. H1N1, which is swine flu. They accidentally grabbed H5N5, which was Spanish flu, and they grew it. And they were working in just normal lab 2 conditions, and guess what happened? Yeah, Spanish flu went into the ventilation system, and an entire floor of CDC was hospitalized. Did y'all hear about that? No, we don't talk about that. Yeah, we learned about it at the American Society for Microbiology Conference. They did not want to admit what happened. An entire floor was hospitalized. Nobody died because people were intubated, they were put on life support, and we had enough machines to get them through. Just like COVID, right? As long as we had machines present, we could help people. But what would have happened if that Spanish influenza had gotten out in the general public? That's the scary thing. We, again, would be running out of life support machines. Because what we have to do is we have to keep you on life support for about 2 weeks, and then it's about time your immune system starts to kick this virus and burns itself out. But if we don't have enough machines, we're in trouble. So it's always fascinating. CDC, they don't, like I said, they don't like to report their mistakes. There are a few whistleblowers. Every conference I've ever been into, there's always been something. One of my favorites was there was a CDC worker who was transporting TB, who got thirsty and went to McDonald's. And so he's in Atlanta. He didn't want to leave his samples out in the car because he knew the TB was too hot. It was like 105 degrees. So he takes his sample tray inside with him, tuberculosis, and they're sealed in tubes, but he takes them inside, puts them down, orders his drink, pays for his drink, and leaves. The sample tray stayed behind. The McDonald's workers are like, what is this? They're like looking, it's all property of CDC. They look inside, there's these tubes. They call the company number. They're like, did you guys lose something? And they were like, Yeah, we'll send somebody back right away. So guarantee that courier is not employed at CDC anymore. Again, did you see that in the news? We don't like to talk about that stuff. Yeah. I just point this out because anybody can make mistakes. And that's why it scares me as a person, a microbiology background, that CDC has smallpox. That's a little concerning because I always get a little nervous. I'm like, we've had a couple of incidences that, please don't practice throwing smallpox. I don't want to go there. Don't leave it at McDonald's. Yeah.
Do they have smallpox?
No, they have